Okay, well, it looks like we've already gotten a lot of participants. We can go ahead and get started. But thank you everyone for coming to our webinar. This is a webinar about gene therapy and specifically the NGGT gene therapy clinical trial. So first we'll be hearing from Dr. Vockley, and he will be just giving an overview of gene therapy and specifically the study. And at the end, we will have time for questions. The NGGT team is also here to answer questions live. So you should have the Q&A box down below. Please feel free to put questions in as we go, and we'll make sure to have time for questions at the end. And also just wanted to comment too. So this study, the reason we're sharing this here on this webinar, this was one that was approved by our registry committee, so through our PKU patient registry. And if you want to find out about more studies that are approved, they are on our website. So with that, I will turn it over to Dr. Vockley for his presentation.
Great, thanks. Let's hope we can make the technology work here. And I'm going to start off by trying to share things. And then I'm going to go into presentation mode. How does that look?
It looks like you need to continue to share, but then it looks good. Yep, there you go.
All righty, perfectly. So thanks everybody for tuning in. I'm Jerry Vockley, and I'm at the University of Pittsburgh, where I head the Division of Genetic and Genomic Medicine. I specialize mostly in metabolic disorders, and much of my clinical research over the years has been in PKU. So it's been very much a pleasure to get to know the NGGT team and have an opportunity to talk to you about gene therapy for PKU and particularly this therapy. And I'm speaking as a site PI for the study. My disclosures here, I've got some research funding and consulting that if you really want to scrutinize it, you can go back and check the recording afterwards. But otherwise, I'm going to move on to the agenda, and I'm going to apologize in advance here. I'm dealing with a little bit of my, excuse me, my asthma, so I might cough in your face for on occasion. Try to keep that from happening. So we're going to start off by talking about what is gene therapy, and oh, wouldn't you know it, I can't silence my phone. I can turn it off. And why we should consider gene therapy for PKU. I'm going to talk to you about the mechanism of delivery of gene therapy, and you see the abbreviation here, AAV. That's a kind of virus that we'll talk about. We'll move to specifically talking about the safety, because I know that that's a huge concern for everybody, including investigators such as myself, in considering a gene therapy study. And then finally, review the therapies, the gene therapies that have been out there thus far for PKU, and come back finally to finish up with the NGGT study. So what is gene therapy? In the simplest terms, it's a way of getting a new copy of a gene or fixing a copy of a gene that isn't working and is causing a disorder in an individual. So in this case, we would be talking about, excuse me, repairing or replacing the phenylalanine hydroxylase gene, the PAH gene. Most of gene therapy in this day and age is delivered by a viral vector. And the vast majority of that is currently focused on something called an AAV, an adeno-associated virus, which is a virus in humans that doesn't cause disease, but can be manipulated to deliver genes to cells. Oh my goodness, I hope I make it through this. And we'll talk a little bit about that process. So why should we consider gene therapy for PKU? You know, there are currently available therapies out there. The tried and true one, which is the most difficult one to follow, and that is a restricted fee diet, trying to keep you from taking in enough fee that or strict enough fee so that your phenylalanine levels stay below the recommended level of 360 millimolar. Um, anytime you have a gene that doesn't work, you can think of it as a block in a pipe. And so in this case, a block in the PAH keeps the phenylalanine from flowing through the pipe to turn it into tyrosine. And some of the ways that we can fix that are by restricting what you put into the pipe. That's what we've already talked about. But you can also try and replace the enzyme. That's a therapy called Palynziq. That you're probably familiar with. That's an injectable enzyme that is able to digest the Phe in the blood and turn it not into tyrosine, but something else that is non-toxic. Excuse me. And then there are other ways of trying to improve the enzyme. That's by stabilizing it, and that's what Kuvan or sapropterin or Cefiance or sepiapterin do. They work by stabilizing enzymes that have variants, changes, mutations in them that keep the enzyme from working properly, and it tickles it into working. Um, However, if that doesn't work, then we're left with trying to replace the enzyme. And with the current technology, the best way to think about doing that is through gene therapy. Um, this targets the underlying, the fundamental cause of the disease by giving you a new copy of the PAH gene and allowing you now to return to being able to change the phenylalanine in your diet to tyrosine. It has the benefit of theoretically being a single dose only. We're not sure how long it lasts. That'll be studies for the future, but it's for years and or decades. And if there's a need to replace it, there'll be something for sure in the future. Uh, AAV has been just around the corner, probably for as long as I've been in this business, which is about 40 years. And the nice thing is that corner's finally here. We've actually turned it. Um, and gene therapy is out there. It's in the wild. There are at least 230 clinical trials with gene therapy. Not all of them in rare diseases. At least 7,000 patients have been dosed with AAV gene therapy as of earlier this year. And currently, there are 8 products involving gene therapy with AAV that are approved by the FDA and or the European Union equivalent, the EMA. And they're listed here. I'm not going to read them all to you, but you can see that the first of them goes all the way back to 2012. So there's quite a long history of using gene therapy in rare diseases, and all the way up to just last year with the approval of a drug for hemophilia B. I'm going to talk about safety first because I think this is uh, the most critical barrier for patients who are thinking about doing gene therapy and investigators who are thinking about offering it. We want to know that it's safe. You want to know that it's safe. Um, and so I'll talk about that a little bit here. Just to orient you, the things that you really want to think about when you're talking or when you're thinking about an AAV gene therapy is as you hear about what's happened in other trials, you have to understand that not all diseases are the same, and that the gene therapy could be targeting different mechanisms of causing problems, and therefore also having a different clinical course. Um, uh, I've already mentioned AAV as a vector, a delivery tool for giving genes into cells, but AAV is not a homogeneous thing. There are lots of different kinds of AAVs, and we'll talk about that in a second. Um, the side effects of AAV are very much dose related, and so you'll see some information about the dosing in this study, and then something that we won't really get into because the FDA sort of takes care of this for us, and that is the quality and purity of the drug have to be really of top at the very highest end, and thus the manufacturing of it needs to be very well controlled, and the FDA makes that an integral part, a necessary part of approval. Um, so what kind of side effects are seen as a result of AAV therapy? There are lots of different things. And they go from actually causing changes in the genetic information in the cell. Some gene therapies have been shown to cause tumors, things like blood vessel clotting, nerve damage or nerve irritation, changes in the brain MRI. But in the context of the vector that we're going to be that's being used for this therapy, it is really specific for the liver, and so the only thing that's relevant for us is what are the liver abnormalities or side effects related to gene therapy. And the main one is pretty much predictable. This is a virus. Even though in its natural state it doesn't cause disease, if you give a whole lot of it and it goes to the liver, it infects the liver cells. It can cause a liver irritation, and cause what's called a hepatitis. Um, that is an issue that's easily dealt with. We can suppress the inflammation that occurs as part of that infection using steroids. And then after that first batch of virus is cleared, then that appears not to be a major problem anymore in liver-based AAV therapy. Um, in the NGGT study to date, all of the AEs, the adverse events that have been reported in the liver, have been temporary. Some of them have resolved without treatment. Some of them have needed some steroids, but they've all gotten better. And so the overall tolerability, safety of the profile is quite good. What could be called serious side effects, mortality is a serious side effect. No one has died from this therapy. Is very, very small, and we're gonna go over that with you now. So if you look at a couple of approved drugs, one called Zolgensma, which is for disease called spinal muscular atrophy. This uses an AAV called AAV9. And AAV9 is a vector that hits all the cells in the body because this is a disease of the muscle and nerves. And so you have to get it into widely into the body. It requires a fairly high dose. And even with that, as of last year, about 3,700 patients have been dosed. It's been very safe. However, there have been two patient deaths, that is likely related both to the systemic delivery and as well as the dose. You see that there are two pluses there. I can get a laser pointer here. You can see right here the dose of this is listed as 2 plus. We're relating that to NGGT dosing at 1 plus. Another drug approved for muscular dystrophy goes in at a much higher dose. 800 patients have been treated, and 3 of those patients have died. And this has actually been pulled from the market at the moment as they look to see how best to control serious reactions to that. Um, as I said, the NGGT vector is directed to the liver. It's a sub type called AAV8, and that one difference makes all the difference. This one only goes to the liver because we're targeting the liver with PKU. The dosing is lower. Now, the number of patients that have been dosed thus far has been much smaller, of course, 21, because we're just in clinical trials, but no one has had a super serious problem, and certainly no one has died. Um, the uh, AAV8 has been used in other metabolic diseases where liver specificity has been important. And you can see a whole bunch of different diseases here that I won't read out to you. But because these diseases primarily have liver effects, AAV8 has been used to enhance the specificity and efficiency of the treatment, and it allows a lower dose with increased safety. The other thing that is possible when you're looking at organ-specific treatments, such as a liver, is you can actually engineer the gene that we're giving back so that it's optimized for expression in that specific tissue, rather than having to have broad specificity across multiple organs. So that also allows you to lower your dose. Um, Looking at other clinical trials thus far, there have been 3, one each by BioMarin, Homology, and Sanofi, and all 3 of those have been discontinued. The reasons given have either been shifting company priorities, or challenges with achieving consistent high-level effectiveness in some of their early studies. So a little bit of a drop in fee, but not low enough and not long enough lasting for the companies to consider moving them forward. Um, that leaves NGGT 002, the study that we're talking about, as the only one right out there right now. And I'm going to come back to show you a little bit more of the preliminary data, patients that have been treated thus far, to support the dosing that's going to be used in the current study, and the results that we've had. And one of the advantages that we have with this therapy is that it has been given FDA orphan drug status, orphan drug designation, which allows it to move through the pathway of approval in a slightly different way that allows it to also move faster. An investigational, they call it a new drug application, an IND, has been approved, and what that means is NGGT is allowed to use this drug now in a clinical trial, and they have been. Um, As you can imagine, don't just go with something like this and say, I just made this drug, I'm going to test it in humans. There's a lot of testing that goes on, first in cells and then in animal models to justify that or to prove that it works, set the dose, and justify moving into people. So this vector has been used to show that it can deliver PAH activity to liver cells. And you can see in the one slide here that uh, where it's been used to treat PKU mice. PKU mice are the PAH mutation is in the background of a mouse that should be black. And so when you get PKU, they get this sort of duller color here. And when you treat them, not only does their phenylalanine go low because their tyrosine can now go to make the pigments that go to the coat color here. These mice now turn black, so you can see these black mice are treated, and their phenylalanine levels shown here in the graph are below a couple of hundred for the most part. So quite an effective therapy. And no really concerns seen in the treatment of these mice. Um, the FDA demands that these genes be studied in non-human primates, and so NGGT has done this. They've shown that indeed the vector goes to the liver in those animals, and you can show expression of the PAH enzyme. These monkeys excuse me, don't have PKU, so we can't talk about efficacy in that standpoint. Um, Now, I mentioned earlier that dosing is everything in gene therapy trials. The lower the dose, the less likely you are to have side effects, and certainly the lower the risk of something as serious as death is you get. And if we just start off down here at the bottom of this scale or this table with the NGGT vector, what you can see is gene therapy trials that have for in various animal studies with a variety of different AAV vectors, even some of them with variants of AAV8. Um, and you can see that every one of these dosing is much higher than the dose that's being used in the current study, going all the way up to almost 1,000 times higher. So we're very comfortable that this much lower dose is going to lead to increased safety in the context of the gene therapy trial. So let's look at the preliminary results that I can share with you. As I mentioned, there are 21 patients that have already been dosed. Uh, the the um, the doses used and these numbers are the number of viruses per kilogram of body weight that are delivered. And so this 6 times 10 to the 12 means it's 6 followed by 12 zeros. That's a lot. But in some of those other studies, dosing was up to as high as the mid 10 to the 14th. So much higher dosing than what you're seeing here. Follow-up has been up to 2 years thus far, and the dosing has been well tolerated with no serious adverse events. I'll show you some of the things that have happened, things that are described as major adverse events that are mild to moderate liver enzyme elevation that has been asymptomatic and transient. And in most cases managed by a little bit of low dose of steroids to reduce the inflammation. What you can see here in this slide is the studies on these two cohorts of patients. One with a dose of 2 times 10 to the 13th, so 2 followed by 13 zeros, and another by 2.5 times 10 to the 13th. And that's it doesn't sound like a big deal. Because this is what's called a logarithmic scale. It is a reasonable jump in dosing, but it's still very much lower than what is being given in any of those other studies. And what you can see here is that at the lower dose, there was effective control of phenylalanine. Here, this dotted line is below 360 for here almost 4 years, but then with a slow increase after that. And so the patients were outgrowing their therapy. But by going up just to 2.5 times 10 to the 13th, now you can see all of the patients in this cohort have stayed not only below 360, but below 180. So they're now all very close to normal in their Phe levels. The other thing that we would like to be able to do for patients with PKU, though, is not just drop their phenylalanine, but increase their natural protein intake. And what you can see here is patients who started off with very low natural protein intake here were able to here with the lower dose still able to increase their natural protein down here in the area where their Phe levels were still below 360. And here's a couple of patients who have one of whom has increased their protein intake to well above the DRI for the daily recommended dose or intake for protein. And so these are still being collected, but it does look like patients are going to be able to, as they stay out here longer term, relax their diet requirements over time. You can see for all of the patients treated, the Phe levels so far. So because a lot of them are still early in the study, they haven't started increasing their protein yet, or they've just started. But you can see all of these individuals out here who are at a year or so are now at above the DRI, and these individuals who are out even further are coming out to yet a higher protein intake close to the natural or to the normal daily intake for somebody who doesn't have PKU. So this has been a extraordinarily successful thus far. If you look at the side effects, what are called adverse events, you can see that one of the patients here who had the higher dose had some nausea, some vomiting that lasted for a day or three, some abdominal distension related to probably the liver irritation for 5 days. These all resolved. Developed some protein and some blood in the urine. Probably not related to the gene therapy, but can't say that for sure. And this is an individual who has maintained their phenylalanine levels at quite a low level here across over 4 years. Has had some intermittent increases of liver enzymes that says consistent with liver irritation, treated with some steroids for some of this, and a little bit of up and down. We don't know what goes on past here, but this is actually a lower Phe level for longer than any of the other trials have ever achieved. Here's another patient. You can see the same dose, 2.5 times 10 to the 13th. Early on, I think I misspoke. Let me just go back to this. This was not a, I was, I said this was a population. I'm sorry. This is that was the previous slide. This is an individual. So you can see the Phe levels of this individual over time. And that's what we have here as well. So very similar looking. The Phe for over 4 years has remained low. Natural intake, protein intake has gone a little higher. This individual has been more cautious in increasing their Phe intake, but with Phe levels down in the normal range, could probably stand to increase even further. That patient had some of the similar things, these early effects related to a large viral dose that has all of which has resolved over the course of a few days. An episode of low blood sugar. Impossible to know if this is due to the liver, to the gene therapy. It may have been related to some liver dysfunction at one of these times when the liver enzymes were going up. But you can see the same pattern: an occasional bump, treated with some steroids, an occasional bump, and now down. So the to summarize what we've already talked about, not all AAV gene therapies are the same. They're disease specific, and the strategies used are specific. The lower dose used in this study translates to better safety, as does the optimization of the PAH gene that's being delivered to have high levels of expression in the liver, that lowers the dose potentially even further. And our ability to target the liver with this AAV8 it keeps you from having symptoms related to other parts of the body. Thus far, one dose has normalized Phe level and offered long-term relief for anywhere between 2 and 4 years. No sign of at the current dose of that waning at all. Excuse me. This just summarizes that in 6 patients, their Phe results, and this elevation of liver enzymes that happens around here in the first 2 to 4 months after the therapy. And we'll explain to you why we will be keeping steroids on during that part of the therapy, should you decide to give it a try. So with that, let me just quickly go over the design of the study. Excuse me. Our inclusion criteria, we can only enroll patients who are over 18, and right now it's capped at 55. This is for individuals who, by virtue of having Phe levels that are over 600 micromolar on their usual diet, will be designated as intolerant, unresponsive, or not able to take other medications. We will test you for the presence of AAV8 antibodies. Some people have those naturally, somewhere about a third, I think it was, NGGT said they were finding. They can correct that in the question and answer session. If you've previously participated in a gene therapy study, you can't do this one right now. And we can't take anybody who weighs over 120 kilos or about 250 pounds. Of course, as in any clinical trial, there's no cost to participate. You'll be brought to one of the centers, put up for as long as is needed for your participation in the various parts of the study, and you'll have all of those expenses paid. Uh, this shows what the study looks like. Each one of those dots is a study visit. Some of them can be virtual, some of them could, or rather, home health. And so you don't have to necessarily be at the study site, but some of them you do have to be there. You'll see that there are four visits. Within the first couple of weeks after dosing. And then they spread out thereafter as you get further along until the 5 year duration of the study, where after a year we'll be following you for every 6 months unless there's a reason to do otherwise. Um, so that first year is pretty intensive. We'll go over this with you in detail if you decide that you're interested, and the things that you probably are used to if you've participated in a clinical trial. Lots of physical exams, blood tests to look at your safety measures, questionnaires to see how you're managing with your disease in the study, diet diaries, and then physical exam for including vital signs as part of the visits. Um, there are currently 5 sites in the U.S. You can see them here in Texas, New Jersey, Pittsburgh, Minneapolis, and Orange County. And you can participate at any of those sites. You probably should choose the one that's closest to you to be most convenient, but you can talk to the study personnel at any of the sites. If you have a particular reason for wanting to go there, like you have family who can put you up, that can certainly be accommodated. If you're interested in participating, here is an email address that you can jot down or get from the recording of this. It is also available on the website of NGGT Bio, which is just nggtbio.com, or you can go through clinicaltrials.gov if you're used to doing that. If you go there and you click PKU and actively enrolling, you'll come up with this trial, and that, or you can enter that number there, and it's there as well. So that's going to be it. I'm going to get out of the share mode here so I can see questions and be able to answer them.
Thank you, Dr. Vockley. Well, great. Thank you for that presentation. And I failed to mention, I know that was a lot of information. As Dr. Vockley said, this will be recorded and we'll post it on our YouTube page. So if you ever want to go back and review it or look up specific information, please feel free to do so.
Very good. And we do have a question online already. That a couple of them that says, "Can I participate from the UK?" Well, you can't participate in the United States. I'll leave it to NGGT to say whether or not they've got a UK site. Yuting, anybody in UK right now?
Oh, I don't know if we can hear you.
Sarah? You can also type it in the chat, and I can read it from the chat if if you're having
Yeah.
trouble
It
getting.
looks like Sarah from NGGT was able to answer. So she said, unfortunately, as of right now, the study is only being conducted in the US, but please feel free to reach out and contact for future information if it expands to the UK or other countries.
Very good. I didn't actually see that, so that means if you can answer those if somebody puts it in the chat.
Yep.
Another question, am I able to participate in other clinical trials should this one not be effective for me? That'll be up to the clinical trial. Most other clinical trials that we participate in, there's a statement in there that says you have to be out of other clinical trials by some specific length of time. Usually it's 30 days. And that's included most of them have included gene therapy for that level of time, or that amount of time. It's the answer is probably, but you'll have to look at each of the trials specifically to know about that.
Dr.
Um,
Vockley, can you also answer if there was a future gene therapy trial, if somebody could participate after participating in this trial?
Also going to be up to that trial. You noted that I said you couldn't have participated in a previous gene therapy trial to participate in this one.
Right.
So the chances are going to be good that early on the answer will be no. But it's a moving field, and my guess is that eventually the answer will be you'll still be able to get gene therapy if and when it's approved or the right vector comes along for you. That's looking into my crystal ball and trying to predict the future for you.
Yeah,
Um,
I think it's a fair statement to say currently with the current research, you would not be able
that
to
is correct.
participate in a gene therapy trial, but
Yep.
down the road, with immunosuppressive therapy, there could be something that would allow you to do so.
The question of how long until this is available for all, assuming the study trials keep going well. Um, NGGT, you want to answer that? I'd say a bare minimum of 3 years, probably a little longer than that, but that's just guessing on the usual FDA pathway.
And by all, I'm assuming you mean people under the age of 18 or over
I
55.
assume they're talking about approval of it, and it'll probably be even longer for children. Thanks for pointing that out.
Mhm.
Yeah, you've got to work your way down to doing something like this after getting it approved or having a reasonable number of adult patients before you can go younger.
Right.
Dr. Vockley, they were also asking if the patients that you're talking about were dosed in the US or were they from previous trials somewhere else?
Most of those patients were dosed in China. There is one patient currently enrolled or has passed screening in the U.S., and I don't think they've been dosed yet. Sarah or Yuting, can you confirm that?
Yes, that's correct. Most of the data shown in the slides are from patients from China, but we have started dosing in the US.
Yeah. Some of my it looks like some of the questions are being answered online, so I'm just going to stick to the open ones. Um, um,
Dr. Vockley, it looks like they want a further clarification of the discussion about getting dosed once for gene therapy or potentially being able to get dosed again. Would you be able to
Okay.
clarify
So
a little
that's
bit
another
more
one of my
there?
crystal ball things.
Yeah, yeah, yeah.
Yeah, at this point, our hope is that it's a one and done. The and that you'll never have to get dosed again. There's a possibility that over time the therapy could wear off, and that's a little complicated. Basically, what happens when this gene gets into the cells, into the liver cells, when the liver cell divides, there's only one copy of that gene there, and only one cell then gets a copy of the gene. Your liver does actually continue to divide and grow and replace itself, and so it is possible that over time the effect is going to get diluted out. Now, you saw that we had some patients that were out of 4 years with no evidence of increase in fee. That could go 10 years, that could go 20 years, it could go lifetime. My guess is it's going to eventually be that the effect will get diluted out. But I'm also predicting that 20 years from now, we'll have way more available for you than we do now. And that's just because I can look back to 20 years ago and tell you that we had nothing for you. And now we've got approved medications and multiple clinical trials that are going on. So the field will continue to expand, develop, and I think you'll get there'll be opportunities to either redose with the same gene if it or the vector if it worked for you once, or go to the next latest greatest vector that's out there if that's what if it becomes necessary.
In that,
Um,
oh, sorry. In
no, go
that
ahead.
same vein, it looks like there's a question. Can you just explain a little bit more why, if someone had maybe already participated in a gene therapy trial, why they might not be able to participate in a different one down the line?
Well, the flippant answer is that those are the rules. We don't make them. To be fair, if you didn't respond to an AAV, the concern is that you've been given a big dose of AAV that you will have developed enough antibodies that you would fight that infection the way you would fight any other infection, and so that it would keep you from having responding to a different gene therapy. Now, there are currently other ways of delivering gene therapy, especially to the liver, that are under investigation that don't require viruses. And so if and when those become available as clinical trials or approved drugs, even if you've had an AAV gene therapy, you will probably be okay to have one of those. There are other viruses that are also being used that don't cross-react with AAV, and so chances are you would be able to get one of those. So remember, we're talking about a very specific point in time. And the answer is no, you won't be able to participate in anything else. But our hope, of course, is that you'll respond. That if you respond, then you won't need to participate in another study because you're going to be like those individuals who are 4 years out and still have fees of 100.
And there's another question, Dr. Vockley, regarding in the U.S. trial, will patients receive the same dose or will the dose be increased in the future?
I'm going to let NGGT answer that. I think there is a potential for dose escalation, but if it's working at this dose, then you don't increase it.
Yeah, I can take that one on. So
Thanks,
we
Sarah.
actually just we just expanded our dose. We just escalated our dose for the next patient that participates in the study. And based on the China studies, this was the most effective dose level and at the lowest point. So as of
Okay.
right now, we're planning to keep that same dose for future participants. And then I
Okay.
also wanted to mention just going off of participating in future studies. I believe the main concern is if a gene therapy trial has AAV-based therapy, then I don't think someone can participate in that after being in this study. But again, like future technology and science may advance and you may still be able to participate in other studies. And then if for whatever reason the treatment doesn't work, people are still able to go back to other medications like Kuvan or Palynziq or something like that. So it's there are alternatives if the study doesn't work, but the goal is for it to be a one-time treatment
Absolutely.
to maintain
Absolutely.
that.
Someone's trying to budge the line and ask about tyrosinemia type 1. Thanks for that question. We'd love to be able to treat all genetic disease. PKU is way more common than tyrosinemia, so it's gotten more attention. There's no reason why these same technologies can't work for tyrosinemia. I'm not aware of an active trial, gene therapy trial for tyrosinemia, I wouldn't be surprised if somebody out there is developing a vector for it.
Well, there's a really dicey question
Which
here,
one?
Dr. Vockley. It says, is there a danger we start to run out of participants for trials or studies if the rules exclude previous patients for future studies? Wouldn't at some point the pool get so small we can't perform a trial or get good data because of such a small sample size?
Well, so at this point, and that is a dicey question.
Yeah.
I'll do my best to answer it. So, right now there are thousands of patients in the U.S. with PKU, and these trials are designed to study dozens of patients. The answer is that if everybody would be willing to participate in a clinical trial, there would be no problem. Of course, you find the right drug, and you don't need any more trials. You just treat everybody, and you're done with it. It's not so much a matter of patients available, but patients being willing. And this has been a struggle for us in all of our clinical trials. I can tell you, our intent is for us to, of course, not just offer it to our active patients, but offer it to those patients who have sort of fallen away from clinic. A lot of our 30 and 40-year-olds who have said, well, I just don't see any advantage to going to clinic anymore. And they're out there with levels of 1200, but are still functioning. It's very difficult to reach those individuals. It's very difficult to get them to come in for clinical trials. I also do a lot of talking at family groups, and trying to get people to reach out to others that they know and say, look, there are these clinical trials. You haven't been in clinic in a long time. You might not know about them. Just in case you're interested, here's a list. And so do feel free to talk to your friends, anybody you've met over your career or your life who has PKU and would be interested in this.
Yeah, and just to put out a word, we totally commend participating if you can, if your lifestyle will allow you. And we thank all of you that have participated in clinical trials, because
Yes.
without your participation, we would not have any pharmaceutical products. And even in the early days, we had clinical trials testing the medical formulas. And many of you have, and I helped in my clinic to ensure that these formulas were appropriate for individuals. And of course, we have our National PKU Registry where we collect natural history data. And that has been super, super informative to clinicians, researchers, industry, to help identify the unmet medical need and what are the best approaches to evaluate different treatments and so forth. So thank you all that have participated in these trials. Thank you for all of you that have participated in our National PKU Registry, because without you and without doing that, I don't think we would know as much as we do in PKU. So just wanted to put a plug there that we really appreciate it. There was another question, Dr. Vockley, and I know it was answered. We have a few more minutes, and so taking advantage of that, that the trial is currently only including 18 years of age and older, and they were wondering why we can't have younger patients in the study. And Sarah did a nice job answering it from a company perspective, but I thought maybe you might provide it from just a clinical trial perspective of why typically
Sure.
they
Yes.
evaluate adults first.
Yeah, happy to talk about that. In both regular therapy and in clinical trials, safety is the most important thing. we would, as physicians, would never bring to you a treatment that we felt was unsafe. Um, and of course, in clinical trials, that safety measure is way lower than it is for approved medications because less people have been treated. And when I say way lower, it doesn't mean that we don't think it's safe. It just means that we want to take the safest possible path to approval. And usually what that is to start with adults. You're older, you can withstand liver dysfunction for a longer period of time until we can fix it if that's what happens, or if something else shows up. You're also old enough to make your own decision. And that's
Yep.
really important. You can say, I don't care about all of those long list of things you put in your consent form, which has everything that's ever happened practically in every clinical trial. They're really daunting documents for the most part. Clinical trials always start with adults unless there's a driving reason to take them into kids. For example, if nobody lives past age 1, well, then you start the clinical trial as soon as you can. Once there's enough experience to show efficacy in 18 and above, then the goal would be to drop it. Most clinical trials have an adolescent cohort, so you go down to about age 12 or so, and then you go down a little bit lower if that's proven to be safe. The other thing to keep in mind with gene therapy is this idea of outgrowing your efficacy. So if you treat a 2-year-old who still has to increase their body size fivefold over the next 15 years of their life, the possibility that they're going to outgrow it is maybe higher than if you're already full grown and all you're doing is replacing the little bit of your liver that gets turned over every year. So there are a lot of reasons why we start with adults and then work our way down, but safety is the absolute most important component of that.
In the last minute or two, I've also seen a question we've gotten multiple times. It's been answered, but just so I'm sure other people have the same question. Dr. Vockley, can you explain if this approach works across multiple genetic variants and maybe how that's different from other gene therapy approaches?
Yeah, so this theoretically should work for anybody. There's some nuances there that if you have a variant, a mutation that allows your cells, your liver cells, to have some PAH enzyme, but maybe it doesn't work very well, there's a chance you're a better chance that your body would recognize this as something that is natural and not attack it. If you've never made this protein because you have mutations, variants in your PAH gene that don't ever let you make PAH protein, your body views this as a completely foreign protein, and it might cause more problems. In reality, that hasn't been that big a deal in gene therapy, and so we don't expect that to be an issue. So the simplest answer is to say this is open to anybody. We don't really think that there's going to be much difference based on what your mutation or your variant is.
Great,
Great.
thank
I
you.
also wanted to add, just because I saw this question come up a few times about the cost to enroll in the study. And for those who didn't have a chance to see the chat, there's no cost to enroll. And we want to support you as much as we can. So we've incorporated home health because the timeline does look pretty intimidating, but if you just take it appointment by appointment, most of those or half of those appointments can be they're just monitoring, and they can be done in the comfort of your own home. We also provide stipends for like compensation for your time if you're missing out on work or time away from your family. We reimburse for traveling, we reimburse for hotel, and we're pretty adaptable to your needs. So we honestly want to work with you as much as we can to make this streamlined and comfortable process for you, if you are interested in
And I hate to do
participating.
this, but believe it or not, I have another one of these starting in less than a minute.
Yes.
So I'm going to have to thank you for your participation and willingness to listen to this. Hope you consider it. If you do have any questions, you want to look at talk about things in more depth, just contact one of the sites that you saw in the presentation, and we will be happy to set you up to speak to someone at great depth about your specific questions for your specific flavor of this disease. And thanks to NPKU for letting us discuss this. I hope that we can bring you something that will help you in the future as a result of this study. So, whoops, I just
Yes.
turned my camera off.
Thank you, Dr.
There we
Vockley.
go.
Thanks, Dr.
I was
Vockley.
gonna wave bye.
Thank
We'll
you.
see you sometime in the future, I hope. Bye-bye.
Thank you, everyone. And we'll post this recording shortly with some of the contact information to contact their study if you would like to learn more. But thank you all for joining. Have a great rest of your day.